Mutation. In biology, a mutation refers to a change in the DNA sequence of an organism's genome. These changes can occur naturally during DNA replication or as a result of external factors like radiation, chemicals, or viruses. Mutations can affect a single DNA base, point mutation, or larger segments of a chromosome. There are several types of mutations. 1. Point mutation, a change in a single nucleotide base, which may result in a different amino acid being produced during protein synthesis. This can be silent mutation, no effect on the protein. Missense mutation alters the amino acid sequence of a protein. Nonsense mutation results in a stop codon, truncating the protein. 2. Insertion, an addition of one or more nucleotide bases into the DNA sequence. 3. Deletion, a removal of one or more nucleotide bases from the sequence. 4. Frameshift mutation, insertions or deletions that alter the reading frame of the DNA sequence, significantly impacting protein synthesis. 5. Chromosomal mutations, large-scale mutations that affect whole sections of chromosomes, including duplications, deletions, inversions, and translocations. Mutations can be beneficial, neutral, or harmful depending on their effect on the organism's fitness. In evolution, beneficial mutations can lead to adaptive traits, while harmful mutations may result in genetic disorders or diseases like cancer. Many diseases are caused by genetic mutations that affect normal cellular functions. Here are some examples. 1. Cystic fibrosis. Gene involved, CFTR, cystic fibrosis transmembrane conductance regulator. Mutation type, typically caused by a deletion of 3 nucleotides, resulting in the loss of the amino acid phenylalanine at position 508. Effect, defective chloride ion transport, leading to thick mucus buildup in lungs and digestive tract. 2. Sickle cell anemia. Gene involved, HBB, hemoglobin beta. Mutation type, point mutation, missense, where a single nucleotide change, A to T, results in the substitution of valine for glutamic acid at position 6. Effect, abnormal hemoglobin, HbS, causes red blood cells to become sickle-shaped, leading to blockages, pain, and anemia. 3. Huntington's disease. Gene involved, HDT, Huntington. Mutation type, trinucleotide repeat expansion, where a CAG sequence is repeated excessively. Effect, progressive neurodegeneration leading to motor dysfunction, cognitive decline, and psychiatric symptoms. 4. Hemophilia. Gene involved, F8 or F9, factor 8 or 9. Mutation type, various mutations, including point mutations, insertions, or deletions. Effect, impaired blood clotting, leading to excessive bleeding even from minor injuries. 5. Tay-Sachs disease. Gene involved, hexosaminidase A. Mutation type, often a 4-base pair insertion, leading to a frameshift mutation. Effect, accumulation of GM2 ganglioside in nerve cells, resulting in neurodegeneration and early death. 6. Muscular dystrophy, Duchenne. Gene involved, DMD, dystrophin. Mutation type, typically caused by deletions or frameshift mutations that result in a non-functional dystrophin protein. Effect, progressive muscle weakness and degeneration. 7. Phenylketonuria, PKU. Gene involved, PAH, phenylalanine hydroxylase. Mutation type, various point mutations leading to enzyme deficiency. Effect, inability to metabolize phenylalanine, leading to toxic buildup and intellectual disability if untreated. 8. Breast and ovarian cancer, BRCA1 and BRCA2 mutations. Genes involved, BRCA1, BRCA2. Mutation type, various mutations including insertions, deletions, or point mutations. Effect, increased risk of breast, ovarian, and other cancers due to impaired DNA repair mechanisms. 9. Marfan syndrome. Gene involved, FBN1, fibrillin 1. Mutation type, missense, or nonsense mutations that affect the protein fibrillin, 1. Effect: connective tissue disorder affecting the heart, eyes, blood vessels, and skeleton, often leading to cardiovascular complications. Mutations causing these diseases can be inherited, germline mutations, or acquired, somatic mutations, and can have varying degrees of impact on health, depending on the specific gene and mutation involved. Mutation is crucial in biological systems for several reasons. 1. Genetic variation. Mutations introduce new genetic variations, which are essential for the process of evolution. These variations contribute to the diversity within populations, providing the raw material for natural selection to act upon. 2. Adaptation and evolution. Beneficial mutations can enhance an organism's fitness, allowing it to survive and reproduce more effectively in changing environments. Over time, advantageous mutations can lead to the evolution of new traits or species. 3. Disease and health. While some mutations can cause genetic disorders or diseases, others are beneficial or neutral. Understanding mutations helps in identifying genetic causes of diseases and developing targeted treatments, such as gene therapy. 4. Molecular biology and biotechnology. Mutations are used in research to understand gene function, create genetically modified organisms, and improve agricultural crops. Scientists use induced mutations to study biological processes and develop new products. 5. Drug resistance. In pathogens like bacteria or viruses, mutations can lead to resistance against drugs, antibiotics, or vaccines. Studying these mutations helps in developing strategies to combat resistance and design better treatments. In summary, mutations play a fundamental role in driving evolution, contributing to genetic diversity, understanding diseases, and advancing scientific research.